So this video is going to be on chapter 14, and we are going to do the first section. It is a huge section, this first section. So let's get started. It's going to be a long one. We need to talk about first sensory perception. What are the different ways that we figure out the senses? What are those receptors in there? So there's lots of different types. We're going to classify them. The first is the free nerve endings. You can see their dendrites are kind of open and dangly like tree roots. And those are, again, if we're structuring them by their receptor types, free nerve endings is one type. The other type is encapsulated, where they look like that tadpole ending on those. And then there are specific specialized receptor cells. And so like the photoreceptors in your retina is an example of those types. So there are three structural types. We're going to classify them: free nerve endings, encapsulated ones that look like tadpoles, and then the receptor specialized cells like our photoreceptors. Another classification: where are they? Are they near the stimulus, which is outside, an outside environment? External. Externals are extero, so that makes sense. If they are near an internal organ in your guts, internal organs, those are going to be enteros. And then if we're talking about a moving body part, those are going to be proprioceptors. I can't get this today. Proprios. So X outside, inside, and proprio is moving. So those are relative to location. And then what about their function, their job? What do they do? What do they sense? So chemoreceptors, just like you would think, are going to detect chemicals. Osmo is going to be solute concentrations. Nociceptors are pain. So I just think of the first part of the word no, you're gonna get hurt. Nociceptors. I know, it's silly, but it helps you remember these things. Detects pain, nociceptors. Mechano were, just like we talked about when we talked about pressure receptors in the skin, and we're gonna talk about those again, are mechanoreceptors for physical stimuli. And then thermoreceptors are just like they sound, temperature. So we're definitely gonna have a lecture quiz question on this, and there will probably be a lecture test question on these different things. So let's get into the different sensory modalities. Again, this section is huge. We're gonna go over all the different senses in this. The first one is going to be taste, gustation. So with those, we need to know about the different papillae. Gustation is sense of taste, and you will have different papillae. Some of these papillae are going to have taste buds in them, and others will not. So you are going to have these big guys in the back are called circumvallats. They're these guys here. These are all circumvallats. They look like pencil eraser shaped. They're big and they're round, circular ones. The circumvallats are in the back. They do have taste buds. Other ones that have taste buds, fungiform, are little round ones, and you're going to find these guys pretty much the whole front half to 2/3 of the tongue are fungiforms. They're going to be paired up with filiforms. These do not have no taste buds on filiforms. These are the thread-like papillae, hair-like papillae. They create friction, kind of like if anybody has a cat. Traveling with our two cats today and the dogs. Cats' tongues are like sandpaper, right? And that's kind of that friction that you're getting there. So filiforms, out of the 4 papillae, they're the one that doesn't have taste buds in them. And then over to our last one is foliate. Foliate look like pages in a book, so hence the name foliate. You're gonna find them on the sides of the tongue. So your filiform and your fungiform are gonna be on the top of the tongue. Foliate's on the side, and in the way back of the tongue are going to be these circumvallats. What are they sensing? Well, we can taste all sorts of different things. Of course, the sweet and the salty, but there is also sour, the acidic flavors, and bitter. And usually bitter are things that are poisonous to us. So we have a lot of receptors for bitter. They're very sensitive to bitter. And then we have umami. Our circumvallats in the back can sense umami, and that's kind of a beef steak, I think is how they phrased it, flavor. If you like rich flavored cheeses, it's that same kind of sense there. So these are the papillae that you need to know, that we can detect all of these different tastes, sweet, sour, salty, bitter, fats, umami. We can detect these things with the taste buds. And how do those taste buds work? Where we're going to have our taste hairs attached to the ends of these guys. And this is our gustatory cell. And so they are detecting that specific thing, whether it's sweet or whether it's salty, but that chemical comes along. And then if that is present, it's going to cause these guys, it's going to cause your taste buds, your gustatory cells to depolarize. And that's going to send a signal to your brain on what you are tasting, depending on what they are sensitive to. So our sweet and salty, our sweet and sour ones are going to, salt is sodium chloride. So they're going to look for sodium and hydrogens, and these are going to be chemically gated proteins, and they're going to open up. Am I going to need you to know the steps of every single process on every single sense? No. Just in general, know that they are going to be chemically gated. Some of them are use a secondary messenger like a G protein, but basically they are designed to detect certain things, salts, sweets, bitter. And when that chemical comes along, it's going to depole that receptor. It's going to depole that receptor cell, and it's going to send a signal to your nervous system saying that, hey, I just ate something deliciously salty, and I love it. So that's how the physiology of it works in a nutshell. Cranial nerves involved in physiology. We talked about it in chapter 13, and we're going to go over them again in chapter 14. Facial nerve, glossopharyngeal, and the vagus are all innervations on taste. So we need to know those. Again, I told you we're going to talk about them throughout the rest of the semester, and we do. So here they are again. Cranial nerve number 7, number 9, and number 10 are involved with taste. Disorders on taste. So if you cannot taste anything, it's agwesia. If you've lost your sense of taste, that has kind of been a COVID symptom, actually. It has been, I'm not hungry, nothing tastes good. That's agwesia. So moving on from taste to smell, olfaction. So these little guys are going to come through the cribriform plate, and you are going to have these neurons. They look like this guy. They're bipolar neurons, and they dangle down through the cribriform plate. The end of the neurons, where the dendrites are going to detect the smells. So here they are, going down through the cribriform plate, and we can see here are the dendrites. And when the chemical comes along, it's going to attach onto these moisture-rich areas, in the top of the nasal area, and bind on there, and those will depole the molecule and depole the receptor cell and send it up there. We will depole these bipolar neurons. They will send an action potential up to the central nervous system, and we will integrate that information, process it, and handle it and detect, associate that with memories. Smelling the flowers is good, or smelling bathroom products is not great. These are the exceptions. So we have talked about in general how usually whatever nerves you're born with or all the nerves you're ever going to get, and they never replace themselves. These are kind of the exception to the rule. So these neurons do undergo mitosis and will replace themselves. The taste buds are not neurons, those are receptor cells that we mentioned previously. So these guys are constantly replacing themselves. Taste buds are always swapping out all the time. The nasal neurons, your olfactory neurons, that go and link up with, where we are, they come down and they're going to go link up with the bulb and then go send the signal through. Those guys are bipolar neurons, and they do get replaced. I'm not going to ask you about the steps on physiology with secondary cyclic AMP or any of this stuff, but do understand that they are bipolar neurons coming down through the cribriform plate, and they dangle down to your nasal passages, and as the air swirls around in there because the nasal concha causes it to swirl. Those smells, those chemicals will attach onto those and send a signal, and we will integrate that in. I am going to switch on down to hearing audio. So parts of the ear are always, they're always questions, either on the lab practical or on the lecture test. It's so hot. 60 degrees in Grand Canyon, and now it is 108 degrees in the shade here in Vegas. It's crazy hot. So parts of the ear, so hot. Parts of the ear are the auricle. So these guys, they are basically like giant satellite dishes. And so the sound waves are going to hit your auricle, and they're going to funnel them into your ear canal. So this is outer ear, your external ear. Sound waves hit the auricle, go towards the hole, the ear canal, and now they're going to travel towards the middle ear. So they're going to go through their external auditory meatus, your ear canal, and which is lined with cells that ceruminous glands, the earwax. They secrete earwax. And the sound waves are going to hit your eardrum, and that is called the tympanic membrane, is your eardrum. And those sound waves are going to hit, and we'll talk about frequency, how close together those waves are hitting or how far apart those sound waves are hitting. But they're going to hit the tympanic membrane. It's going to cause it to push in. And so with the tympanic membrane, we are officially in the middle ear, going through the ossicles. There is the malleus, the incus, and the stapes or stapes. Those are your ossicles, and they're going to transmit the sound to the cochlea, and we're going to go through that process. But these are the basic parts here. External ear, middle ear is going to be the inside of your tympanic membrane, malleus, incus, stapes, your ossicles, and then we go into the inner ear. The inner ear is going to have the cochlea, the vestibule, and any of the membranous bony labyrinth going on there. So here's another close-up of that middle ear with the ossicles. We have the malleus, the incus, and the stapes. That's the sound in which they, as the sound waves hit, that's the steps, who gets hit first. So the tympanic membrane moves every time a sound wave hits. It's going to move, and it's going to push on the malleus or the hammer. The hammer is going to hit the incus, which is kind of like an anvil. The hammer is beating the anvil. And then the incus is going to be attached to the stapes. The stapes looks like stirrups. So those are your three ossicles. That's the order in which the sound waves hit. Tympanic membrane, malleus, incus, stapes. And the stapes is going to then push on something called the oval window, which is attached to the cochlea. There are some muscles. There's the stapedius muscle that's going to take care of the stapes and make sure that it stays where it needs to be. And then we have the tensor tympani muscle, and this guy is going to pull our malleus and kind of keep it tight in case you ever get exposed to really, really loud sounds. It pulls it tight so you don't damage your ears. Inner ear, that's going to be the cochlea, vestibule, semicircular canals. So bony labyrinth, when we talk about those semicircular canals, that's these guys. So we have the bony labyrinth, semicircular canals, and all of this stuff in yellow here. On the inside is going to be the membranous labyrinth, and that's all of the semicircular ducts, which are in blue here. Then these guys are going to connect to structures, utricles, and saccules that are filled with fluid inside, and these are going to help us with balance. We'll talk about balance in a little bit. And then, of course, another part of the membrane is the cochlear ducts. So membranous labyrinth, semicircular ducts, all of these guys here, and this guy out here. And then our utricle and saccules are these guys here. So what happens when the sound waves hit? Sound waves come across, they're going to hit the tympanic membrane, just like I was telling you, and it depends on the frequency, how fast those sound waves come. But they're going to hit the tympanic membrane, it's going to push through the ossicles, step number 2, to displace the ossicles, then the stapes, the last ossicle, is going to push on something called the oval window on the cochlea, and that is going to move the fluid inside of that ear, the perilymph, to move the fluid down the canal. And when it finally gets on the right frequency, we have sensors inside, receptors inside, depending on different frequencies. We can only hear from X range to X range. And those frequencies will get a trigger and set off a signal. So sound waves come in, they're going to push through, and those sound waves are going to go travel across something. It's called the organ of Corti. Here's our sound waves. They go all the way up the snail shell, and they'll come down the snail shell. You can see it pushing up, and then it'll trigger it, and sound waves will come down. And how they exit is called the round window. So we have, here's a close-up of the organ of Corti, and it goes in between. So we were saying that the sound waves are going to travel through and go up, and then they're going to come back down a different way. So as the sound waves are coming through, here's our organ of Corti right here, and it's got this tectorial membrane that sits on top of these basilar membrane that are covered in cells. So let's get a close-up of those guys. All right, so here's our tectorial membrane. It sits on top of these little cilia, these hairs, and that come out of the hair cell. And depending on the frequency, it's going to push down on that membrane, and that's going to move the hair cells, move the cilia that triggers the hair cells, and lets a person know that they're hearing at that specific frequency. So here it is at rest, the hairs are standing straight up. And here it is when sound waves come through, it pushes down on the tectorial membrane, which is all part of the organ of Corti, and it sends a signal that that frequency, that specific spot, got triggered. And so we can hear this traveling through depending on what the sound waves are. So there's two terms that we need to know in this picture here. We have frequency. I keep talking about frequency. And frequency, if you look at these guys, they're a wave, right? It's just like visual light, the sound waves. They're traveling in waves. There's going to be a trough, and there's going to be a crest at the top, and a trough and a crest. Where are those wave crests? Are they far away, or are they close together? That's your frequency. If they're really close together, you're going to have a high frequency or a high pitch, a squeaky sound. If you have a low frequency, those guys are spaced far apart, and you get really low sounds. So a low pitch. So it's all about the wavelength. Amplitude is how loud I'm talking, or am I talking really quiet? That's amplitude. So amps are measured in decibels, and so they have this handy dandy chart here where you can see about how loud things are, and will it potentially cause hearing loss. So we can see, normal traffic, air conditioner, how loud an air conditioner is. Busy traffic, that's about I swear, like the sound here in Vegas is really loud with all the air conditioners running and construction and stuff. It's loud. So I'm probably exposed to, 70 decibels. If you were riding on the subway, if you were operating machinery, you can see that the decibels get louder and louder. And you can, if you're exposed to that for, in this case, it says more than 2 hours, you can risk permanent ear damage from these things. So it's good to know. I had a professor back in my day that he used to go to the gun range. I was in wildlife, I'm a wildlife scientist. And so all my professors were hunters. And he would go to the gun range and he didn't wear any ear protection. He didn't wear any ear protection. So by the time he was, 50s, he had lost a significant amount of hearing because of the sounds of the gunshots, making him suffer some hearing loss. So we can see the nerve is our here it is, vestibulocochlear nerve, cranial nerve number 8. And we've talked about the cochlear part, the hearing part. What about the vestibulo part? That's going to be our balance. How do we keep track of that? So with balance, we got static, that's just me sitting here in this chair fighting against gravity. Or we have dynamic equilibrium, and that's going to be movement when you're fighting against a movement, whether it's centrifugal force or maybe you're riding in the car. We have two different types of equilibrium that you will keep track of. And how do you do that? You're going to use that bony labyrinth and the membranous labyrinth inside of there. And inside of there, we said that those semicircular canals met at this place, and they had the utricle and saccule. And those guys are filled with the lymph, but they're also filled with these super cool structures, and they have crystals inside. There's otoliths, and the crystals are going to sit inside of this kind of jelly-like substance. So you've got crystals sitting in jelly, and those are going to sit on top of hair cells. So it sounds familiar, right? We got hair cells in the ear, we have hair cells in the semicircular canals, in the utricle and saccule, hair cells. So just like before, if the hair cells move, it's going to send a trigger. If the hair cells are straight, then everything's fine. And how does the hair cell move? It moves when these crystals stuck in this jelly-like substance, which is called a statoconia. When they move, because your body moves, because you're exposed to different things. So static equilibrium, you're just sitting here. And then what if I duck my head real fast? If I duck my head real fast, I'd look like this guy. So the force of gravity is going to move those crystals stuck in that jelly-like substance. It's going to move the statoconia down, and that's going to tell those hair cells, going to trigger the hair cells and let my body know that my head moved down. Or maybe I move my head back like this. It'd be the same but opposite movement, right? But it's the purpose of the hair cells to do this. Here's a close-up again of that membranous labyrinth. This is what helps us directionally. So we have these canals, and we have one going this way, and one going this way, and one going this way. And we can tell, you know, up or down, right or left, depending on these guys. So these are going to help with dynamic equilibrium. What happens with these, with our semicircular canals, we have the endolymph, the fluid that's in there, and it's going to move, and if it moves, it's going to trigger the hair cells just for static equilibrium, and those guys sitting on the jelly in the statoconia, here hair cells in them. And when we're at rest, the hair cells stand up. When we move, the fluid pushes the hair cells. So every different canal, one direction, a different one, in a different direction. So it lets you know right or left, up or down, so on. And if you were doing you were going to go in circular movements. This here is tucking in tight, and she's spinning faster and faster and faster. So that is going to flow in the opposite direction of where she's moving. So it's flowing in this way, I'm moving in this direction. And then when she stops, when she sticks out her arms, and she slows the movement, when she stops, that moves in a different direction. So if any of y'all have ever done this, maybe you were just, I don't know, on field day, we always had field day games at the end of the school year. And so maybe at the end of our school year, you had to do where you put your head on and you sit there, then you had to spin in a circle, spin in a circle, spin in a circle. And then you stood up, and then you tried to walk in a straight line. And it's because of this. It's because your endolymph was going in one direction, and then you stopped it, and went in a direction to set off. So this is dynamic equilibrium when you are moving your body in different positions. Again, it is cranial nerve number 8. And this is what it's about here. We have deafness. It could be conduction or sensorineural. Conduction, where it sounds exactly like it's just something's in the way. Maybe you have too much earwax. And then sensorineural would be damage to the nerve cells. If you have otalgia, you have ear pain. Do you have infection? Do you have otitis media? No, external. Maybe you have lost the ability to have your sounds. That is this guy. Pretty presbycusis, and so my professor I was telling you about, he couldn't hear sounds on the birds or making sounds. We're trying to identify them, he couldn't hear them. So he was suffering from presbycusis. And then there's tinnitus. Anybody ever, I don't know, been exposed to that? I've been to a club or been to a concert and the music was really loud. Just about ringing in their ear. That's tinnitus. And vertigo is when your balance is off. Why is being so slow? We have talked about screen is we've talked about these guys, our sensory receptors in the skin, free nerve endings of the epidermis, tactile discs are a little bit lower in the dermis, and ascendians are way down low in the dermis. Refresh yourself on those basic lecture tests, but we've already gone through them, so I'm not going to spend time go through them again. But you know them, they're in the outline already. I'm going to just skip on exercise. Visual is going to be the last part of this sight, and so we need to know the parts of the eye. So we have the eyelashes, they're actually from the outside, which are your eyelids. You are going to have your cornea, which is the clear window into your eye. That lets the eye in, the light in, and it is going to be surrounded by an iris. Iris is the colored part of the eye. I'm not going to ask you lateral and medial canthus, so there's the corners of your eye. Yeah, let's keep going. We do have the muscles. You will need to know the muscles on the eye. I'm hoping that this is recording. It's really slow. One muscle that we haven't talked about though, we talked about the rectus muscles and those pulled straight back. And there was also this guy, the levator palpebrae. The levator palpebrae is going to lift up your palpebrae, so this muscle lifts up your eyelids. We've already talked about the cornea, and then there is the conjunctiva. Cornea is the clear part on the front of your eye, the window. Pretty much if you scratch your eye, you scratch your cornea. And it is connected to the rest of the eye, conjunctiva. Skipping on past it. If you have an infection of the conjunctiva, conjunctivitis, anybody's ever had pink eye. It could be bacterial or viral, by the way. Your extraocular muscles, we already know these. So no results. As far as the lacrimal gland, that's going to produce your tears. So they're going to form in the lacrimal gland, and it has antimicrobial properties, and that is going to produce the lacrimal, like your tears. The ducts is going to slide across your eye as you blink. You're going to move your tears across the eye, and then it's going to go down the whole lacrimal punctum that are connected to the canaliculus, the little canal, then go to drain down into your sinuses. And so if you have ever gotten, missed in your eye and were trying to be a big girl, wearing big pants and not cry, and you were blinking down your tears and you start to sniff. So it's like they sit down your sinuses and start to sniff. Right, so past the cornea, you are going to have the iris that we talked about. The pupil is going to be the hole where the light goes through. And then it is going to refract off of the lens and go back. There are three tunics in the eye. There's the fibrous one, that's the outermost one. It's going to have the sclera. The sclera is the white part of the eye. And then we've already talked about the cornea, the clear, transparent part of the eye. Behind that, we're going to have the anterior portion of the eye that's filled with your aqueous humors. Then let's go to the vascular tumor. That's where you have your iris, the colored part, whether you have brown eyes, blue eyes, green eyes. And it's going to have your ciliary light, and that's going to have muscles. It's going to attach to your lens. And it's also going to be a brown pigmented layer. And that brown pigmented layer is going to be important when we talk about how the light is attracted to this dark brown pigmented area. And then we have the neural pigment is going to include your retina, So it's going to have your rods and cones. Your cones are going to sense color. The rods are going to be black and white, just light and dark. So those are your different tunics. Cornea, iris, going to hit the lens. Let's refract the light towards your retina in the back. Anterior chamber is filled with aqueous humors, which are more liquid when we compare it to the posterior chamber, which is filled with jets of humors, which is more like jello consistency. We haven't talked about in this image. The canal of Schlemm, we said that the anterior chamber is filled with liquid. Sometimes that liquid pressure, otherwise you get glaucoma. And so the canal of Schlemm is going to allow for that pressure to escape that area. What else have we talked about? We talked about the ciliary muscles. There is the ciliary process that's going to add onto these suspensory ligaments, and the suspensory ligaments hold the lens. And so when the ciliary muscles are relaxed, skip ahead, when the ciliary muscles are relaxed, the pupil is dilated. When they contract, the pupil is smaller. And what that does to the lens, when it relaxes, the lens is flat like a pancake. When they contract, the lens gets squeezed and it's more round. It changes the light on, the way the light refracts off your lens. These guys, yeah. Dilation, pupil is wide open. Restrictions, pupil is smaller. And then we already talked about the neural part that's going to have the retina in the back. That's what we talk as far as anatomy. It's going to have the fovea and the macula lutea. And so you're going to have your highest visual acuity in this area. Your fovea centralis is going to be only cones. And the macula lutea is going to have cones and rods. And then as it spreads out from there, it's going to be a mixture. Further out, lateral aspect of the eye towards the outside of the eye, the more lateral you go, the more rods you have, the more needle-like, the more cones you have. So here's the fovea, macula lutea. The this is your blind spot. That is where you don't have any sensors to detect the light because your optic nerve goes out there. The nerve number 2 is running out of that, and you don't know what's going on there because the space is already taken up by the nerve. How does your eye detect the light? So light's going to come in, and remember I told y'all about correlative pigment, dark color. So we are going to be, the light is going to be attracted to this dark pigment, and as the light gets attracted to the dark pigment, it's going to set off your rods and your cones. So these guys are going to sense different frequencies of light, and then send that signal with your bipolar cells to your central nervous system. You know what it is that you're seeing, what colors are you viewing, how bright is the light or how dark is the light. It has gotten sunsets down here, and so it's gotten darker, and my rods are telling me this, that the light is darker now than it was earlier. My cones would be telling me about color. Fovea centralis, the high visual acuity, has more cones, and then further away from there you're going to have more rods. And in between is going to be rods and cones. Here is, again, in the Pearson image, again, high density of cones towards your fovea. As you go away from there, with the cones. Rods are going to be highest on the outside and lowest on the inside. So they're kind of opposite. Wavelengths of light, your visual light spectrum, you can only detect certain wavelengths because those are the receptors that our body is geared with, and we can only detect from this frequency of light to this frequency of light, these wavelengths are what we see with our eyes. Other animals can see other frequencies. We cannot see ultraviolet light, for example, but other things can. Bluebirds can detect ultraviolet light. Owls can see ultraviolet light. Butterflies, not butterflies, bumblebees can detect ultraviolet. As the light comes through the lens, it's going to refract. Talked about sympathetic and parasympathetic. We kind of touched on it, but we want to take it out in systems. And for sympathetic activation, we are going to have that light refract differently on the lens. The lens is going to be flat. The ciliary muscles are going to relax. The lens will be flat, and the light is going to refract back onto the back of the eye, onto the retina. What gets sent back there is inverted. Inverted is upside down and it's backwards. Everything that gets sent back there is inverted, and it's up to your brain to switch and flip that image to make it right. Your brain is going to fix this for you. So when it integrates this information, it will change it. It will show that on the back of your eye. It's just like it would be on a microscope. It is inverted. It's upside down and backwards. So this is your sympathetic is built for distance, looking at things far away, because back in the day we weren't always the top predator. We evolved with other predators, and so we need to be able to see those guys at a distance. Is the lion on the plains coming to eat me? So this, we need to see things at a distance. Is the sympathetic activation. The lens is flattened, and it helps us see at a distance. The ciliary muscles are relaxed. Parasympathetic. Sympathetic was your fight or flight. Parasympathetic is your rest and digest. So if I'm digesting my food and my dinner is here and I want to go and eat it, I need to be able to see what I'm eating and making sure that I'm eating good for me things, and maybe I get a poisonous berry or something. So I need to be able to look and focus up close. Parasympathetic. So in this case, so here, let's contract. The lens is going to bulge, so it's going to be squished up, and we are going to look at things close up. So just like we said, all of the images are getting refracted to the back of the retina, and when the image comes through, it is inverted. Your normal vision, if you've got 20/20 vision, you have immetropic eyes, you've got perfect great eyes. Good for you. Most of this, you could have nearsightedness, myopic, to where you can see objects close, like we were talking about with our parasympathetic. You can see things up close just fine, but at a distance, you can't see them. So you can correct these guys. We can correct nearsightedness and farsightedness with corrective lenses, glasses, or contacts can help us with this. And it's because, why can't you see this? With somebody who's nearsighted, your point where the image is refracting off and then it's going to come together, it should be hitting right here. Your fovea centralis should be here. Remember, that's your point of highest visual acuity. But in this case, that's not it. Your fovea centralis is way back here. Your eyeball is too long. And so you need to use corrective lenses to make sure now, with the corrective lenses, that it does go back to the fovea centralis where you need it to be, and it fixes itself. So this is myopic nearsightedness. There's hyperopic, and that's farsighted. Then processing. Remember I said that stuff that comes in on the right side gets processed on the left, and stuff that comes in on the left side gets processed on the right. It's true. It happens with your eyes. And so you're going to take your information through your optic nerves, cranial nerve number 2, and it's going to go back. They're going to meet at the optic chiasm, and then from there, the fibers are going to split. And so all of our red fibers are going over here, and all of our blue fibers are going over here. And so we're going to process on different sides, different hemispheres, what we are seeing, and then we'll put that information together. I've got a really cool video in chapter 15. It's relatively long, maybe about 5 minutes, maybe 10. But it's really cool because it talks about a person who was having seizures so badly that they cut his corpus callosum. So they severed the connection between the right and the left brain. And so when he processed things, he looked at them totally different. And so he would process a telephone as a totally different word when he was trying to write down and describe what he was seeing. So it's kind of cool the way we process stuff, that some things go to the left side and some things go to the right side, and then we have to put it all together and figure out what exactly we're looking at. I'm not going to make you diagram out exactly what goes to what side. Lots of disorders of the eye. So obviously we've already talked about nearsightedness, we talked about farsightedness. There is astigmatism, that's what I've got. And so that's where your curvature of your lens is just off. It's not right. So either the lens or the cornea is off, and so it's causing the light to not hit in the wrong place. Glaucoma, we mentioned before, where you've got that intraocular pressure, that interior area is filled with fluid, and it's under too much pressure, and that leads to glaucoma. Diplopia was the double vision that we talked about. You could get, if your cranial nerves are acting up, you could get double vision. We talked about other cranial nerves acting up could give you strabismus. Your eye could rotate external, or your eye could rotate internal for cross-eyedness. Color blindness is when your receptors, your cones on your eyes, they're not processing the colors like they should. So there is red-green color blindness. It is linked to the X chromosome. And so that is passed to males and females, right? But only males have how many chromosomes? They only have one X, right? So if a male only has one X, he's more likely to have color blindness because it's passed on the X chromosome. And you could not see cataracts. You could not see this. You could not make out this number here. If you were red-green colorblind, it would all look the same to you. You wouldn't be able to see the number. Cataracts, where the cornea is clouded up. And the nyctalopia is our last disorder in this whole thing. And that is where it is hard to drive at night. My mom does not like driving at night anymore. It's a result of a vitamin A deficiency, but it's kind of like, you know, when you were in a bright room and you went into a dark room and you didn't turn on the light switch, you know, it takes your eyes time to adjust. So it's a lack of being able to adjust to that, to be able to see in low light, is nyctalopia. And that has to do with the vitamin A and being able to process and turn over those signals quick enough. So this is the end of this video. It was a really long video. And then I'll come back and do another video later on the rest of chapter 14.